Welcome to lecture second of cell biology. In first lecture we have studied the syllabus, what is syllabus prescribed by the university. Now the next part, that is the prokaryotic cell. Myself Dr. Sunil Patil from LVH College Panchoti, Nasik. This is lecture second of cell biology. So prokaryotic cell, pro means primitive, karyon means nucleus. Nucleus is primitive without nuclear membrane, so they are called the prokaryotic cell. All living organisms are grouped into two main categories, prokaryotic and eukaryotic cells. The definition: pro means primitive, karyon means nucleus. Organism in which the nuclear material is not bounded by definite nuclear membrane are called prokaryotic. The nucleus is not bounded by definite nuclear membrane. Example: bacteria, blue-green algae, mycoplasm, PPLO, pneumonia-like organism. The nuclear material commonly consists of single chromosome living in the cytoplasm called as the nucleoid. This is the circular DNA actually. Prokaryotic cells are found in bacteria, cyanobacteria, that is blue-green algae, archaea, etc. The structure of prokaryotic cell: first we are starting from the outer side, that is covering, called as the cell envelopes. The outermost is glycocalyx, it is outermost capsule or slime layer, or it is also called as the extracellular polysaccharide, gelatinous polysaccharide and polypeptide covering or lying externally to the cell wall. So it is outer to the cell wall. Some bacteria have loose sheath called slime layer, with other may have thick or tough sheath called as the capsule. So this glycocalyx may be slime layer, may be capsule. The capsule may protect pathogens from phagocytosis and enable adherence to surface, prevent desiccation, may provide nutrients. So these are the layers shown, three layers of the cell envelopes of the prokaryotic cell. So this is prokaryotic cell. You can see the structure. The next or middle one is the cell wall. It is rigid. It is middle coat. It is made up of peptidoglycan, or called as the murin, in eubacteria, as well as pseudopeptidoglycan in archaeobacteria. It maintains the overall shape and size of the cell. The inner to cell wall or innermost is called the plasma membrane. This is lipid bilayer similar to the plasma membrane of eukaryotic cell. This layer transport protein, waste, ions and nutrients across the membrane. So you can see these three layers. Outermost is capsule or slime layer, middle one is the cell wall, and innermost is the plasma membrane. The next part is the mesosomes. Mesosomes, a special membrane structure formed by extension of plasma membrane into the cell, called as the mesosomes. So you can see the extension inside the cytoplasm of the inner plasma membrane. It is called as the mesosomes. You can see in this figure also they form a vesicle. In this way they done the process. The mesosomes either tubular, either vesicle, or either lamellar. They form tubule-like structure, they form sac-like structure, they are branch sac-like structure called as the tubule, vesicle and lamellae. These are three types of mesosomes. They help in cell wall formation, DNA replication and distribution to daughter cells. They also help in secretion processes, respiration, to increase the surface area, etc. In some bacteria like cyanobacteria, they are membranous extension, they are colored, called as the chromatophores with pigments. So these are the three types, and in cyanobacteria they are the chromatophores. So these are the mesosomes. You can see in the figures, these are photographs where formation, deep formation of tubule-like mesosomes, etc. Then we are moving to the appendages, means outer structures. The first important one is the flagella. Flagella are long filamentous appendages, consist of filament, hook and basal body. Filament, hook and basal body, and help to push the cell rotating. Motile bacteria move by taxis. Then axial filament. The axial filament are similar to flagella except they wrap around the cell and spiral cell that move by means of axial filament, endoflagellum. They are found in the spirillum, means helical bacteria, as they are also called spirochetes. So you can see the structure of axial filament. Then femori and pili. The femori and pili are short appendages. Femori are usually many and the cells adhere to surface. It is a function. Pili are 1 or 2 or up to 4 also. They help join cells during conjugation. You can see in figure the many in number, short, they are called the femori. Few, these are yellow color, they are called the pili. They are used for special type of movement called twitching. In Pseudomonas, in Neisseria gonorrhoeae, and some strains of E. coli or gliding movement of Myxobacteria. So you can see this femori and pili in the figures. It is the photograph. So we are moving to the internal structure of prokaryotic cell, that is the cytoplasm. Cytoplasm is fluid. The fluid part, which is cell surrounded by the plasma membrane. The cytoplasm is composed of mostly of water along with organic and DNA, organic molecules, DNA, ribosomes and inclusions. The nucleoid is the part. The prokaryotic DNA, called as the nucleoid. The DNA associated proteins are not found. So it is only DNA, not called as the chromosome, and complex nuclear membrane are not found. Occurs are, it is called as the nucleoid or nucleoid region. So single circular DNA is present inside the bacteria. These are the photographs. In bacteria, nucleoid associated protein NAPs interact with prokaryotic DNA and function like histones. Instead of histone, there is presence of nucleoid associated protein of eukaryotic cell assisted in the organization packaging of the chromosome. Ribosomes. The cytoplasm of prokaryotic cell contain numerous ribosomes of the 70S type. Large subunit is of 50S and small it is of 30S. Togetherly, sedimentation coefficient decreases to 70S. where S stands for Swedberg. Ribosomes are made up of ribosomal RNA and protein, rRNA and protein, and serve as the site of protein synthesis. The plasmid, it is one thing, ribosome, nucleoid we have seen, then plasmid. Prokaryotic cells are also contain small circular double-stranded extrachromosomal DNA. They are called the plasmid. Plasmids are generally many in number per cell and often carry genes for the advantageous traits such as antibiotic resistance. Plasmid mainly function as antibiotic resistance. In addition to the circular nuclear DNA, the plasmids are DNA structures. They are small circular double-stranded. The ribosomes, we have already seen about the ribosome. So this is the structure of the plasmids, and this is nucleoid region. This is the plasmid and this is the nucleoid region, single circular DNA, and this is the plasmid. You can see other structure. The fifth structure, it is the inclusion. Inclusions are reserved deposits found in the cell. Some examples of prokaryotic cell inclusion found in the bacteria are metachromatic granules, polysaccharide granules, lipid inclusion, sulfur granules, carboxysomes, magnetosomes, and gas vacuoles. Sixth one is the endospore. It is different thing. Endospores are resting structure formed by the bacteria like bacilli and clostridium during unfavorable environmental conditions. In unfavorable environmental condition, they go to the resting phase and they call it the endospores. The process of endospore formation is called the sporulation. The return to its normal state when environmental conditions are normal, the vegetative state called as the germination. They become active, called as the germination. So these are some structure. This is process, vegetative cell, they goes to the endospore formation. So social environment. prokaryotic can inhabit nearly all environment. Some archaea and bacteria can even thrive in extreme conditions such as temperatures, they are called the thermophiles, or highly salinity called the halophiles. Temperature, thermophile, high temperature when they survive, and in high salinity they are called the halophile bacteria. Such organisms are togetherly called the nextremophiles. Some prokaryotic live in one or more on the bodies of other organisms. They are called the symbiotic prokaryotes. Many archaea grow as plankton in the ocean. Then reproduction in the bacteria and archaea reproduce through an asexual mode of reproduction known as binary fission. During binary fission, the genetic DNA undergoes replication followed by division. That replicated DNA divides. Site selection, then invagination of the cell envelope and synthesis of new cell wall in at the middle. The division takes place. You can see in the figure there is single circular DNA which replicates. It is shown in two color. Then a depression occurs and two cells are formed as a normal binary fission. Two cells are formed. In this way, reproduction takes place in the bacteria. So this is about the bacteria. Then we are going to see about the E. coli. That is Escherichia coli. It is a prokaryotic cell, a typical prokaryote. Escherichia coli, commonly called the E. coli, that is gram-negative, facultative, rod-shaped bacteria, about 2 micron long and 0.5 micron in diameter. You can see the photograph of E. coli. Optimum growth of E. coli, it is at 37 degrees centigrade, that our body temperature, but some laboratory strains can multiply at the temperature of 49 degrees centigrade also. It is commonly found in lower intestine and of warm blooded, including man, so they are called the endotherms. Most E. coli strains are harmless, but some very few, these serotypes are causes food poisoning also and are occasionally responsible for dysentery. The harmless strains of this E. coli are part of normal flora of the gut. Means in our gut normally the E. coli are found. They can benefit it to us host by producing vitamin K2. The E. coli produce vitamin K2 helping us or helping to the warm-blooded animal, and by preventing the establishment of pathogenic bacteria. Other bacteria cannot be established as there is presence of E. coli bacteria inside our intestine. So these are some photographs and the structure of E. coli. Cells are able to survive outside the body or are limited for limited time, which makes them ideal indicator organism to rest of the environmental samples. So we can test it by testing the fecal content. E. coli is the most widely studied prokaryotic model organism. It is important species for field of biotechnology, microbiology. It has served as the host organism for majority of work of the recombinant DNA technology. So rDNA technology, the E. coli is most commonly studied. So these are links I have used for this lecture. Thank you. For next lecture we will meet again, that is for the eukaryotic cell. That will be third lecture. Thank you very much. See you again.